It's Melissa and Katy from Selling Again. We have our season 1, episode 4 of Last Week in Stem Cells, and if I'm being honest, it was kind of a slow week. Although we have at least one story in here that I'm really excited about.
Guess which one it is for points. What is that show where you can get all the points and it means nothing, or
all the points means nothing?
No, you can get any points you want, but they don't really mean anything.
I think that's life.
No, it was like it's a stand-up comedy show. Oh, well. Anyway, so that's beside the point. So our first story coming to you today, is from a scientist at the University of Buffalo that created a non-toxic method to produce stem-like cells using mechanical stress. So the research was made possible by genetically encoded optical probes. Probes.
Don't say it again.
No. The blank. The probe measures the stress and actin within living cells. So what is actin? Let's do a refresher. So actin is actually a key structural protein in cells, and it's involved in a lot of different cellular processes. So it's like in cell signaling and shape and movement, and it's especially important for muscle contraction. So the actin probes provide key information in real time about how mechanical forces influence living cells, tissues, organs, and eventually animals. So they created these transgenic worms and flies that express the probes. Ooh, creepy crawlies. And the researchers have demonstrated that their method was non-toxic to cells. So therefore, it could be used to study in whole animals. So
I feel like this sounds too good to be true, because every time someone says we found a new really easy way to make stem cells, it turns out to not
like
work.
The STAP method. Have you guys been paying attention?
Like STAP.
So now it'd be the probe, the probe method.
So our next study is on using platelet-rich plasma for total knee arthroplasty, which, if you guys are not familiar, that's a knee replacement, giving you bionic legs.
They just hide it in some fancy words that, it can just confuse you.
Yes, they hide it in fancy words so you don't know that they're turning you into part robot. Which, frankly, I mean, I'm kind of excited about that. My grandma has 2 total knees, and I think she's indestructible. So anyway, this study was looking into using PRP on patients that were undergoing total knee arthroplasty. And what they found, interestingly, was that the PRP gel did not seem to significantly improve pain immediately after surgery, or to provide really any benefit, which, if you think about what's happening in a total knee arthroplasty, that kind of makes sense, because you're replacing a bunch of biological stuff with mechanical stuff that doesn't necessarily have a lot of biological signaling components, so PRP might not be the best fit there.
So our next study comes from the Colorado Blood Cancer Institute in Denver.
This is the one I'm so excited about.
Oh, you gave it away.
Oh, my bad.
So this team from Colorado published their findings in, I don't know, is it JAMA Neurology? JAMA?
JAMA, yeah. Journal of the American Medical Association.
JAMA, Bobama. All right, I probably shouldn't say stuff like that. Well, anyway, they had 24 study volunteers, and they went. These people had MS, multiple sclerosis, and they underwent some stem cell treatments between 2006 and 2010. So most of these patients that took part in this study are still in remission several years later. And the phase 2 study has demonstrated impressive results by rebuilding the immune system using a patient's own stem cells. And the scientists thought that perhaps the new immune system would no longer see the myelin covering of the nerve cells in the brain and spinal cord as a threat. So if the stem cells could halt the myelin attacks, the MS disease process would be stopped.
It's a really cool study, because they basically went, hey, you know what? Your immune system is doing something it's not supposed to do, so we're going to hit Control-Alt-Delete and just start over.
That's usually how it works sometimes.
That's every day at work, yeah. Okay, so this next one is interesting for all of you guys that, we get a lot of questions here about, is fat ready? Are fat stem cells happening now? And so this company...
Give me the skinny, Katy.
Exactly. Give me the skinny on fat. So this company, Antria, they are developing these adipose-derived stem cell treatments for what they say is a number of different pathologies, although the one they happen to be going after first is plastic surgery type applications. And so
people want to look younger.
They do, and they're willing to pay for it. So it looks like their secret ingredient, they say, is based on collagenase, but Melissa and I suspect that it's just collagenase. So
what is collagenase? Remind us, Katy.
Good question, Melissa. Collagenase is an enzyme that will break down the collagen fibers in fat, which kind of makes it disintegrate, which releases the stem cells that are in the fat so that they can be used for, in this particular case, injection under the skin, kind of for the treatment of like wrinkles and, to make you look younger and prettier.
So why would, sorry to interrupt, but why would people or the FDA have any concerns with collagenase usage?
Well, collagenase, according to the FDA, using collagenase is above the line of minimal manipulation, which, if you guys are reading Ted's blog, which is available at sellingbiosciences.com/blog. // My bad. Don't use a backslash. Ted talks about that a lot. Anyway, so long story short here, Antria is actually pursuing an IND, so they're getting this approved as a biologic drug, and they are moving into phase 2, which is pretty interesting, and, it looks like they're the ones that are the furthest along with this type of an application. So, good news for everybody who wants to use their fat to make themselves pretty.
Bring on the burgers. All right, our next study shows some insight into motor neuron disease using stem cell technology, and it was published in Nature Communications. So scientists at the University of St. Andrews have discovered new ways of studying what happens to motor neurons affected by motor neuron disease. So what they do is they get some patient skin samples, and they manipulate those cells and turn them into stem cells, and that is how they kind of experiment with them to see how normal cells would react under this disease, and how and also disease cells would work as well. So the scientists say why motor neurons are lost in MND, which is motor neuron disease. They think that using new developments in stem cell technology would enable them to compare the function of motor neurons from healthy individuals with those from patients suffering from different forms of MND. So there we go. Much better put together science.
All right, this next story is one of the places where it seems like manipulation of autologous cells might actually be beneficial. For the most part, we here at Selling Biosciences think you can accomplish probably anything you want without manipulating your cells outside of the body. But in this case, people have been really enamored for a long time with this idea of taking stem cells and then injecting them, and then them being these smart little injury or illness-seeking machines that float through your body and just go fix what you need fixed. And that would be... So that would be a really good idea if it weren't for the fact that they all kind of get stuck in your lungs. And if your lungs are the problem, then great. We've delivered them efficiently there, but in terms of doing, full body circulation of mesenchymal stem cells, it's been kind of problematic. So this study showed that you can take mesenchymal stem cells, and instead of growing them where they stick to a surface like plastic, they took them and put them in a solution, like in a liquid, and kind of rotated around so that they never get to stick to a surface, and that made them smaller, and that was small enough that more of them could just circulate through the lungs instead of getting stuck in them. So it'll be kind of interesting to see where this particular technology goes, and if they find out that this is a legitimate reason to manipulate cells outside the body, if in fact you can inject them and then they turn into little nano machines that go in and do all of your regeneration work.
There's always hope.
There's always hope.
So,
what are y'all doing?
Hello, Ginger.
Don't see it.
Say hello.
Ginger just walked into our office, but we're going to keep going because we've already made too many bad jokes in this episode.
So our last story is on embryonic stem cells. So when embryonic stem cell research first started becoming really popular, before all the ethical stuff came into play, people were wondering, well, what if I just inject a embryonic stem cell into the body? Why won't it just do what it's supposed to do? And what ended up happening was, like, it was starting to create basically tumors, because if you think about it, an embryonic stem cell is supposed to grow into a person or an animal or whatever, a complete structure, and it receives all those signals once it's in the womb of the mother. But when you take it out of there, all those signals get really mixed up, and it ends up kind of just, creating tumors. So people were trying to figure out, well, how is it that we can harness the power of the embryonic stem cell? And some researchers at the University of Copenhagen actually found a way, or actually had a comprehensive analysis of a pathway that was important for these embryonic stem cells to divide, and it's called ERK, E-R-K.
Mm-hmm.
I feel like it should have a better name. What do you think? But
I like ERK.
You like ERK?
I do.
It's like Joe Schmo. ERK. You kidder, ERK. But as a result, they basically found a way, to block this ability. So if the embryonic stem cells can't really differentiate and they just stay as embryonic stem cells, you can really keep them there to discover more things about them. So, yay, ERK!
Kind of like when I was little, and my mom used to say how she wished she could keep us from making bad decisions, like jumping a bike off of the roof into the sand pile. If she could have blocked those choices, she would have. And this is the same thing,
Nice.
We're just blocking their choices and controlling them and keeping them from becoming horrible, horrible cancers.
Oh. My mom wanted me to grow, so that was kind of the opposite, but it never happened. So. Sorry, Bob. Legs didn't get the signal.
I'm sure they'll have a pill for that soon.
All right, thanks everybody for putting up with our silly jokes. Happy Monday.
And if you want to follow us on Twitter, she's at Cell Whisperer.
And Katy is at Dr. Lightning Texas, TX.
And the company is at Selling Bio.
Woo! All right.
Y'all have a good week.
Good night, everybody.